Okay. Well, welcome everybody. Still got some joining, but let's kick off. We're going to run this strictly to time. My name is Charles Nicholls, and I'm delighted to have so many of you here today. We're basically going to review a tiny little bit of detailed introduction into the charity, the carbon community, and what we're trying to do. And then look at this exciting new study that we've just published. So from the study design to first results, and Dr. Bonnie Waring, who is Associate Professor in Ecosystems Ecology at Imperial College. Wave, Bonnie, say hello. Is We'll be presenting that. Then we're going to go over to a panel discussion lunch, is panel in just a moment, and then Q&A. Q&A is always some of the best bits of these kind of events. So please do ask your questions in the question Q&A section if you can. It's better than in the chat. They're much more likely to get picked up. If you have a question for a particular panelist, then please do direct it and we'll try and steer it to the right place. If we don't get through all of the Q&A, then what we'll do is we'll try and answer as many of those we can afterwards. Okay, so with that, let me introduce our panel. Our panel, actually in alphabetical order, represents, together with Bonnie and one other, our scientific advisory board, and our scientific advisory board steers the charity and the research in terms of where we should be going in terms of future directions. So from the top, Colin, who originally designed some of the study, and in fact was one of the first people I talked to, and out of a conversation came a crazy idea. So thank you, Colin. And he was at the time at the Crowther Lab at ETH Zurich and is now working in the space of microbial enrichment in forestry in the US as CEO of Funga. Professor Martin Bidartondo, who's a professor of molecular ecology at Imperial College and honorary research associate at the Royal Botanic Gardens, Kew. And then Professor David Beerling, who's director of the Leverhulme Centre for Climate Change Mitigation and Professor of Natural Sciences at University of Sheffield. And last but not least, Professor Pete Smith, who is chair in plant and soil science at the University of Aberdeen. So that is your panel today. I'm honoured to be amongst you, but let us kick straight off into what we are really trying to achieve. So 2 slides really before we get into the meat of this, just on who Carbon Community is and really what we're trying to do. This is a relatively young charity, about 6 years old now, which brings together sort of principles of sort of trees, science, and people, if you want to put it simply, where we focus research specifically really to try and figure out how we can use nature-based solutions to accelerate carbon removal in trees and soil. And the results we're presenting today, or Bonnie is as a result of our first paper, which really looks at the above ground data. So we all know here that much of the carbon is stored below ground, and obviously here we're primarily looking above ground. So there's more to come, but in terms of first results, this is sort of kind of very interesting in what we see. The tree science and people part is that the charity runs a large-scale field trial site in mid Wales, and Bonnie's going to talk a little bit about that. And then we have dedicated scientists working on that, funded in part by the charity. And the people part is the citizen science. And in this study to date, the first 4 years worth of data, we had almost 300 people involved in capturing tree measurements and tree data. And some of you are in the audience, I know, and I want to thank you on behalf of all of us, really. This isn't possible, without citizen science contribution. The audience today is quite a mixture. So we're going to go from a fairly high level down to hopefully quite a low level, a deep level, depending upon the questions that get asked. Everything from members of the public, donors, supporters in other forms, citizen scientists, our scientific team, and quite a few other universities and forestry organisations around the UK and in Wales. So thank you all for joining. When we started the charity, we sort of had this big idea, which is very simple really to say, and very hard to execute on, which is, can we accelerate carbon sequestration in trees and soil? If we're going to put trees in the ground, how can we do it in such a way that we can help nature along with nature-based solutions to accelerate carbon sequestration? It sounds simple, but I'm now going to hand over to Bonnie, and she's going to talk you through what is arguably one of the most complex field trial sites to try and answer these questions anywhere in the world. So Bonnie, over to you.
Thank you so much. So I'm really pleased to be here and excited to share the results of our first paper in communication sustainability, which came out a few days ago. If you can advance, please. So this undertaking is enormous in its physical scale and in the amount of effort that was required to gather the data. We trialed 2 different interventions to enhance tree growth. One was adding rock dust, which I'll discuss more in a moment, and the other was adding beneficial microbes to the roots of the trees at the time they were planted. We did this to 2 different types of tree communities, single stands of Sitka spruce and a diverse mixture of broadleaves. And this combination of treatments, which yielded 8 treatment combinations in total, was replicated 8 times across a landscape the size of 18 football pitches. And you can see the schematic of our experiment here. Next slide, please. So what everyone wants to know is, what was the answer? And our big result is that we were able to increase the amount of carbon stored in wood by between 13 and 27%, depending on the intervention that we applied. This result is for small trees. This is reflecting data collected by year 4. So we think that if these treatment effects persist, the absolute amount of carbon captured could be very large in the end. But we're seeing uplift in carbon sequestration of nearly a ton per hectare in the best treatments. So how did we achieve this? Next slide. Next slide, please. So again, we are looking at 2 interventions applied to 2 different types of trees. We had Sitka spruce, which is intended to be a representation of what we would see in a forestry context. And we planted a diverse mixture of native species, which is intended to emulate natural woodland restoration. And I'd like to emphasize that for most of our results, the biggest impacts were seen in this broadleaf species mixture. So that's where our potential gains are greatest. Next slide, please. So the first thing we did was apply rock dust, trialing something called enhanced rock weathering. There is actually a secondary pathway by which this treatment can capture carbon dioxide from the atmosphere. But what we found is that unexpectedly, this rock dust stimulated the growth of trees by up to 27%. That's a massive number, which would make any forester really happy. So particularly in our deciduous mixture, we saw this massive gain in the growth of individual trees and in the amount of carbon that the whole stand of trees accumulated at the plot level. Next slide. Rock weathering is essentially an attempt to accelerate a natural process which is already removing 100 million tons of carbon from our atmosphere every single year. Basically, as very common types of rocks called silicate rocks weather, they react with rain and with carbon dioxide, and that chemically transforms that carbon dioxide into something called bicarbonate, which is a dissolved inorganic form of carbon that can be exported to the ocean and can store the carbon there for thousands of years. We accelerate this process really simply by crushing and grinding the rock, which just makes it react faster. Next slide. So the initial impetus for this treatment is that we would accelerate this process simply by introducing the rocks not only to the rain and the atmosphere, but also to the roots of trees that would accelerate the rock dissolution and this overall export of carbon from the atmosphere into the ocean. Next slide. We applied crushed rock twice, once in 2020 before any trees were planted. That was an extremely simple intervention that mimics what we would do if we were doing enhanced rock weathering in a cropland, which is where it's mostly been explored so far. And we applied it again in 2023 when we did have trees growing on site using a modified lime spreader. Next slide. So again, what we were really looking for was atmospheric capture of CO2 via this direct weathering reaction. And we did see this, although at a very slow rate. But what was more surprising and encouraging is that this rock dust also increased the soil pH, and it itself contains nutrients that the trees took up. We could see this, it was reflected in the nutrient concentrations in their tissues, and that's responsible for this growth boost to the trees. And this is really exciting because it means that if we deploy enhanced rock weathering in a forestry context, we actually have two paths of carbon removal from the atmosphere, one through the weathering reaction itself, and two, through this growth stimulation of the trees, which we think should work anywhere where the soils are deprived in the nutrients that the rock contains or where the soils are acidic. Next slide. Our other intervention was adding beneficial microbes to the trees. And this was a very simple intervention. We didn't culture anything. We weren't buying a commercial product. We simply visited a nearby forest that already had growing healthy trees, took a scoop of soil, and added that to the tree's roots. And when we did this, we saw that on average, the trees tended to store a bit more carbon, about a 13% increase. Although I should mention that at the whole plot scale, this was not statistically significant. Next slide. So again, this intervention was relatively simple. We were just adding soil directly to the roots. This was a one-time intervention conducted at the time trees were planted. All of the spruce trees received soil inoculum from a spruce forest, and all of the broadleaves received inoculum from one single broadleaf forest that happened to be dominated by oak trees. Next slide. The idea behind this intervention is that trees need friendly microbes to grow. And the types of microbes, particularly the fungi that associate with these trees, are ectomycorrhizal fungi that simply do not exist in grassy habitats like the sheep pasture that this site was before we initiated the planting. So essentially here, we're not only restoring the trees of the forest, but we're restoring the below ground community of the forest as well. And work by Martin and his team at Kew has shown that there was a high diversity of these ectomycorrhizal fungi in the roots of the trees that were planted. Next slide, please. So here is the overall summary of the results that we found across our project. On the left-hand panel, you can see the percentage increase in the size of an individual tree stem as a result of these interventions. So trees that received the microbial inoculum grew on average 5% bigger. Those that received the rock dust grew on average 9% bigger. The right-hand panel shows how these results at the individual tree level stacked up to the carbon stocks in the whole forest stand on a hectare basis. So by the end of 4 years, the trees had about 2 tons of carbon per hectare, 2,000 kilograms of carbon in their biomass on an aerial basis. And we increased this to about 2,300 kilos per hectare in those that had received inoculum, and nearly 2,500 kilograms of carbon per hectare in those that received the rock dust. Again, these carbon numbers are small, absolutely, because these are very young trees. But if these treatment effects persist through time as the trees grow much bigger, so will these carbon effect sizes. Next slide. I'm showing you here some information about how these treatment impacts changed over time. So again, on the left-hand panel here, I'm showing you the percentage effect size due to the rock dust and the inoculum treatment in each year that we measured tree size. So you could see that in the first year of the experiment, this inoculum treatment actually had a massive influence on tree growth. Those that were treated were nearly 50% bigger than those that were untreated, and that impact dissipated over time, whereas the rock dust effect was more constant. And that's probably because, as the right-hand panel shows, the pH impacts of the rock dust were also fairly stable through time once the rock dust was applied. Next slide. And finally, something that might be particularly interested to those of you on this call who are managing forest yourselves. I mentioned that although we saw an increase in tree growth with the inoculum treatment, when we summed it up and looked at the whole plot level, the carbon storage difference wasn't significant. Why is this? It's because in our inoculum treatment, the impact of that treatment depended a lot on which species you looked at. So in the worst case, we have the alders, which actually didn't show any growth response to the inoculum, whereas the oak and spruce trees really, really liked this treatment. And we think, again, that's because the soils that we used came from an oak and spruce forest, respectively. So that's really showing the potential that with more refined matching between the source of the inoculum and the tree you're planting, there's the potential for big growth gains. Meanwhile, for the rock dust, we didn't see as strong a species-specific treatment. All of them benefited more or less equally. Next slide. And I just want to say that although these bar graphs are relatively simple, they reflect an enormous input of work and time and effort from a huge team of volunteers, over 200 of you, who measured repeatedly 6,400 individual trees. That's 75,000 individual measurements over 4 years. And this is just the beginning of the story. So I'm sure many of the questions we'll get in the Q&A will be about how these treatments evolve through time, and we can speculate, and that's really important for us to know. But the only way we can know that is to continue these measurements, which we can only do with your help. And so we on the science team are extremely grateful for that. Next slide. So to summarize the results overall, we developed 2 relatively simple, low-cost interventions that had demonstrable impacts on tree performance. The addition of rock dust increased tree growth by up to 27% in the broadleaf specifically, with an additional modest amount of carbon capture directly through the reaction of this rock dust with the atmosphere. We also showed the potential for a growth boost with the microbial enrichment treatment, 13%, although this didn't achieve significance across all the species, but the direction of the species-specific responses shows us that we can probably fine-tune this treatment and have really good outcomes. So overall, this site already, only 4 years into its lifetime, has captured excess carbon dioxide, and we're really looking forward to carrying this work forward and seeing how we can improve upon it and extend it elsewhere. So thank you so much for your time, and I'm happy to answer questions.
Fantastic. Thank you very much, Bonnie. So before we go to the Q&A, please do put your questions into the Q&A panel, and we will come back to those in just a moment. I'm going to go to each member of the panel and just get their observations, because one of the great things about our scientific advisory board is we have real breadth here. So covering different areas and different disciplines. And to some extent, you see that reflected in the study itself. It's a combinatorial study where we're combining different things together to then try and find solutions. So I'm going to go in reverse order from alphabetical. So I'm going to start with you, Pete. Pete, do you have any quick thoughts or observations really on the study, and maybe the things that surprised you most or you found most intriguing?
Yeah, so firstly, I mean, the fact that this got off the ground is the most amazing thing, because we're used to doing this sort of experiment. Well, I say we're used to. This sort of experiment is fairly rare when you run it as a research project. So for this site and this experiment to be set up by a charity and to be run by a charity and to draw in all the expertise that you've managed is absolutely exceptional. So it's been really great to be part of it. What surprised me, I think, is seeing the impacts of both of the treatments. You know, the inoculants and the basalt, showing that there is some impacts on the trees, and that shows that even if we're not interested in carbon dioxide removal, if we're not interested in the climate change impact, it actually makes good sense in terms of silviculture to apply these things. So it's got a number of implications, not just for climate change, but also for silviculture and for growing trees. So that's my main reflections. And a fantastic study. I wasn't a co-author on the study, so I can say this independently. It was really well conducted, excellently written up, and it's great to see the publication out.
Brilliant. Thank you, Pete. David, over to you.
Yeah, I endorse what Pete said. I think it's amazing work that you've done as the carbon community to get it off the ground. I think what's exciting is really the fact that it's a large scale trial done in the real world, not in pots in a greenhouse. It's incredibly well replicated, so that when you see the conclusions, you can have confidence in the responses you've seen. I think in many ways, really exciting to see the basalt improve the tree growth, and it supports some of the work that was done in the 1930s by European foresters. There were one group in Germany where they showed that actually, poor management of forests had led to a decline in soil health, and they added basalt, and that restored soil health, and the trees grew better. And another one in Czechoslovakia or the Czech Republic showed that SO2 from power stations is also causing acidification of soils and limiting tree growth. Adding basalt restored tree growth. So it's the first time that I've really seen a modern study rigorously conducted that shows basalt can improve tree growth. And of course, it has very exciting implications for reforestation projects for climate change mitigation.
Thank you. Martin, any observations or thoughts?
Yes, hello. So yeah, I second what Pete and David said. The scale is what really blows us all away. Really, we are often doing studies that are large scale surveys or small scale experiments, but this is sort of a combination of both. In that, in the early days when Charles and Heather and Colin were designing this, that the scale is really what really set this apart. And so it's been a great opportunity to dive into the microbial side and the soil and the roots into in a study that is done at this scale and that has real world implications and great potential for following it further over time. We are currently looking at the roots and analyzing that with a PhD student, and that's turned out into quite an interesting detective story. And also for further work or trying to replicate these kinds of analysis in other parts of the country, perhaps. So yeah, very exciting stuff.
Okay, and Colin, any thoughts?
Yeah. First of all, just so happy to see this, really hitting this major milestone. I mean, I remember taking the call with you, Charles, many, many years ago, probably, right at the beginning of the pandemic, maybe, or before.
Yeah.
Yeah. And, just throwing out this idea and just, seeing your ambition to go big was the most exciting thing. Um, a lot of my expertise is in microbiome restoration, so I can comment directly on that. We're seeing really interesting responses. I think the species level variation is most interesting. Some of these tree species are very responsive and others less so. And the other thing I want to emphasize is that, when we did this inoculation, it was during the pandemic when these trees went in the ground, and we had to take, which forests do we have access to, without knowing necessarily a ton about the background microbial composition of what went into the study. And even with that sort of very coarse shot on goal we took, we got responses. And so I think as this type of intervention becomes more sophisticated, people are doing a lot of work and learning that the donor environments you use really matter. So a forest is not a forest is not a forest. Even single species stands can have wildly different sort of mycorrhizal communities. And so but regardless, like even the signals we're seeing here suggest there's a lot of promise here. And then lastly, the scale is really what matters. Most field studies like this are literally a 20th or the 30th of the size of this experiment, right? They'd probably represent 2 or 3 of the 72 experimental plots we have. So just to give you a sense that scale is so important. We're talking about ecosystem ecology. This is truly manipulative ecosystem ecology, which is rare in my experience.
Yeah, thanks, Colin. And you say that we had ambition to do scale when we first had a conversation. It actually grew through the discussions with you and David, and in particular when David said you really should try these two things together based upon the body of research. We went from 36 to 72 plots overnight, and of course that made it way bigger. And in fact, I should just give a quick shout out to Alan Wilson, who I think is in the audience. Sorry to call you out, Alan. But you did the original mapping from what Colin designed, in sort of squares into actually field structures. And I know you've moved on now, but I just wanted to sort of recognise that you're part of the original team too. So let me go back to Bonnie first. Really with a follow-up question, which is, so where does this go next? What are you most excited about, and what can we look forward to? Having got these results, what new avenues now open up?
That's a great question. And I think every single panelist talked about how the size of this experiment was one of its most exciting features, but I'm going to say we need to go even bigger. So it's really exciting that we show that this experiment works at this particular site. But ultimately, we want to know if this works at most sites, or which sites these interventions might be most effective in. And the only way we can answer that question is by conducting similar experiments elsewhere. Now, that doesn't mean that we need 72 plots, at this huge scale, but to tactically conduct similar tests of these interventions in places that are chosen. So they tell us, can you still get this basalt effect in a higher pH site? Is it really the more degraded soils that benefit more from the microbial enrichment? And so by pairing that with the results of this study, we can start to develop essentially a guidebook for land managers who are choosing which intervention to apply. The other thing that's really exciting, of course, is carrying this forward through time, because as Colin said, these are ecosystems. The stand dynamics will change through time. The trees will grow. They will also encounter more climate stresses like, the one we've been having over the past months. And that gives us the opportunity to see how our treatments influence the long-term trajectory of these forests. So bigger in space, bigger in time. I think this experiment could have a lot of value for the UK forestry community for years to come.
Okay, great. So great shout out there upon satellite or replicas. I mean, for people in the audience who are planning on putting the forestry in, then please have a conversation. We'd love to talk to you about how we might be able to replicate some of this. David, let's come to you next. Thoughts on the future?
I think, as Bonnie said, what's crucial is getting a long-term picture of how these trees respond. They're only young, so they're in an early growth phase. So you really, in an ideal world, would like to carry on for another 5 or 10 years, funding constraints permissible. I think that's crucial. And then the second thing is really to understand what's going on below ground, because there may be a strong root response to the addition of the rocks, and you might have a much higher increased root biomass, which at the moment is still kind of under investigation. If you want to build the complete sort of carbon budget of the ecosystem, you really need to know what's going on below ground. So I think my two sort of top things to investigate would be, how are these responses maintained, or how do they continue in the longer term, and how do we drop a complete carbon budget for the ecosystem?
Hey, great, thank you. Martin, you want to go next?
Yeah, sure. So I agree with all of those ideas for future exploration. As I said, we are very keen on trying to understand what's going on, or what has been happening in that site with the roots themselves. So that's kind of the next thing that we're trying to sort out. I think it's quite clear that the mycorrhizal type of the trees is a big deal, and that really does make a difference here. So your rowan and your cherry are in a different world really than the oak and the birch, for instance, because they just don't share the same fungi. And that has taught very different implications for how these trees grow and how they get nutrients and how they even interact with this addition of the rocks. So that is definitely worth looking into. And I think another thing that we've discussed with Colin actually in the past is the matching up not just the trees with the fungi, but also the source. Where are the best places to get the most beneficial, impactful microbes to add in correct combination to the trees that you're planting. So that is very much an area for exploration and discovery, I think.
Right, great segue. Perhaps Colin, do you want to go next?
Yeah, I'll second much of what Martin just said. Seeing, this replicated in space in more locations is the name of the game, right? We know just so many things vary location, whether you're talking about environmental conditions, land use history, underlying soil types, all of this matters. And then second, what we call the donor environments, where you source those native fungi from, can have wildly different communities of fungi, which can therefore drive wildly different effects. And so the more we can replicate this, the better we can understand, really, much much of this research is actually inspired what people have done and by what people have done in medical microbiology. So your gut microbiome has this profound impact on your health. And, we know you can take, the right gut microbes from a healthy person and use those to treat a sick person. This is the treatment for chronic C. diff infection. And you need to take them from the right person. You can't just take them from anybody. And so I think there's a lot of exciting work to be done in the optimization of those sort of donor forests. Like, what are the donor forests that are harboring the outstanding fungal communities? We've been doing this a lot in the southeastern US at our company, and we probably have 100 trials on the ground at this point, much smaller than these, but testing many more donors. And there's substantial variation in response to those microbes. And so the more we can, discover, what are the habitats holding on to really interesting microbiology, the more impact potential I think we'll find in the UK landscape.
Right, thank you. And Pete, looking to the future, what are your thoughts?
So it's trees take a really long time to grow, right? And what we're describing now is just the first couple of years. So I'm really interested to see what happens as we move forward into the future. Do we just give trees a good start and then the other ones catch up? Or does that good start that we give them benefits all the way through their life cycle? So that's what I'm really interested in, seeing the interactions and just seeing how the two treatments, the mycorrhizal fungi and the basalt, act in their individual cases, and just seeing what happens over time, because really ecology is a long-term process, and hopefully we can keep this study going for a number of years, and that's when we'll start to learn how long-lived these effects are. So that's what I'm really looking forward to.
Yeah, absolutely. Thank you. And I will add one that I thought was going to come out and didn't quite, which is actually the antagonistic effect, where we layered both the microbial enrichment and the basalt together. Didn't have the expected outcome, but in its own way, that opens up a whole new avenue of research to then try and figure out exactly what is going on under the ground, and it very much moves into a below ground story as we were talking about earlier on. So first off, so thank you to the panel. This has been really, really helpful and useful. I'm going to open it up to audience. If you would like to pop some questions into the Q&A, then please do. And I see that Pete's already got in and answered one already, but I'm going to read it out because I think it's interesting for everybody else, which is, how do you evaluate the amount of carbon stored in wood? Bonnie, you actually did the work, so do you want to jump in on this one?
Sure. So we took individual measurements of 6,400 trees. This was basically the diameter of the trees. And foresters for a long time have been chopping down trees of various sizes and literally weighing them. And from that, we develop an equation that tells us if a tree is this size in diameter, it will contain this much mass. And luckily for us, the amount of carbon per unit mass is very standard. So we use what are called those allometric equations to do all the carbon calculations. But it all hinges on the actual size measurements that the volunteers have taken.
Yeah, many thousands of them at this point. Yeah, we've all become expert at measuring baby trees. Okay, next question. What pH is appropriate for the addition of basalt? So I think we're talking about soil pH. David, do you want to have a go at this one?
Sure. So maybe Bonnie could say, what was the pH of these soils? I think
5.5 to start.
Sorry, go on.
5.5 to start.
Yeah. So anywhere in sort of 5.5 to 6.5, 7 pH range is good. You wouldn't particularly want to do it on alkaline soils, although it's not unheard of. The benefits for the trees really come from the increase in pH as the basalt weathers, which allows the trees to take up nutrients more effectively. And the other thing to mention is that, as the basalt weathers, it releases nutrients that support tree productivity, like phosphorus, potassium, and some calcium, magnesium. And so the more it weathers, the more the roots and the mycorrhiza can respond, and the more the trees can hoover up those nutrients.
Yeah, absolutely. Okay, where in the UK is the addition of basalt most appropriate? Anybody who wants to have a go at that?
I can answer, and I think it links to another question I saw about the Q&A about what type of rock is appropriate, and also how much it costs in carbon to do this. So any silicate rock can produce carbon capture via the weathering effect and stimulate tree growth. However, you generate a lot of carbon emissions moving that rock from the place it was produced to where your trees are. So the optimal place to site this is in a location that is already near some sort of mining operation where the basalt dust is generated. And in general, the more calcium and magnesium that rock contains, the better it will be for the trees and for the other route of carbon capture. So there's lots of places, particularly in the west of the UK, that fulfill all those criteria.
West of the UK and Scotland is also where most of the tree planting happens too. So yeah, so proximity to a basalt mine, that's important. You mentioned also other types of silicate rock. David, do you want to jump in on that one? Is this only basalt, or can you use or any type of rock? Can you?
Yeah, well, basalt is probably the optimal one because it has these accessory nutrients and it's very abundant. It's widely mined. Just to go back to the earlier question, I mean, the scientific community is not just looking at adding crushed rocks to reforestation projects. It's also focusing quite strongly on the co-application of basalt with agricultural land as well. And as you said earlier, you do want to minimize the distance between quarries and fields. But because of these properties for restoring soils, it does have potential for carbon capture on agricultural land, increasing food security and reducing our reliance on supply chains for fertilizers like P and K, which mainly come from Ukraine. So it reduces our susceptibility to geopolitical shocks for the farming community.
Okay. When will you have more information on the carbon sequestered below ground?
Very soon. We already conducted our survey of root biomass and soil carbon. So we measured soil inorganic and organic carbon in every single plot before we did anything in 2020. And then we took soil cores and analyzed them again in September of 2025. And actually, the final sample was analyzed about 6 weeks ago. So that data will all be coming online relatively soon. We also have a paper in review looking in more detail about the carbon capture via the weathering itself and what happened to all that inorganic carbon. And so that paper should be out relatively soon. This first paper took over a year in review. Most people know how that goes, but the data is on its way.
Very thoroughly reviewed then. Yes. Okay. Right. Anyone have any more questions? Then do pop them into the panel. That would be great, and we'll get those in front of our group. Anyone else on the panel have any observations they would like to make at this point? No.
I did see a question about why the two treatments together didn't work that I thought was interesting, and maybe I could expand upon and others could provide some input, because this is when we originally designed the experiment, we had a combined rock dust and microbe treatment because we thought they would actually complement each other. So the microbes that we're adding might basically weather the rock dust faster and produce a virtuous circle, basically. And we found the opposite. So why is that? We can't say for sure, but one thing that we did notice is that when we added the microbial inoculum and the rock dust together, we got a completely distinct soil fungal community to what we saw in either treatment alone or the control. And so what this suggests is that perhaps when you raise the pH or modify the soil as you do with the rock dust, that is not liked by the microbes that we're adding in our inoculum. And even more interestingly, the microbes that we added in our inoculum seem to slow down the weathering of the rock dust. So there's some kind of antagonism going on there, which is really, really cool, actually, because it means that if we understand better the way that microbes are interacting with these rock particles, we actually could design, treatments that complement each other. And it shows the importance of biology for this overall weathering story.
Okay, thank you. Yes, one area I'm absolutely fascinated by is that interaction. Unexpected results always are intriguing. Another question in from the audience. The color of the trees in the photos looks very healthy. Did the site receive standard ground preparation before planting, or was there additional cultivation? So this is kind of a forestry kind of question. Ground preparation was using a continuous mounder. There was nothing else done effectively other than what you would consider to be normal forestry operations, using a mounder, planted by hand, etc. So the only other treatments that were applied were then the soil injection and the prior application of basalt before the mounding took place. Okay, so hopefully that will answer that one. Which of these techniques are relevant to conifer timber plantations? Who wants to have a go at that one?
Well, I'm happy to jump in. So in this particular experiment, we looked at one conifer timber species, which was Sitka spruce, and it didn't have a pronounced response to the basalt treatment, unlike all of the broadleaf ones. And it did experience some minor benefits from the inoculum, but it didn't translate to a big increase in wood growth by year 4. So why is this? It might be because this particular species prefers a more acidic soil pH. We actually didn't see an increase in calcium or magnesium concentrations in its tissues. Does that mean that this wouldn't work for any conifer? No. There has been work out of some research groups based in Belgium that are looking at historical rock dust applications to timber plantations on extremely acidified soils that have been subject to acid rain. And in those experiments, there has been a major growth boost. So, if you're considering this, it might be worth considering what your soil nutrient profiles and pH look like. And we think that probably you could get a benefit under those more acidic conditions.
And Bonnie, maybe just jumping on top of that, I mean, what about sites where you might expect higher mortality? If you've got a problematic site, would you consider that as a potential candidate then for one of these treatments?
Yeah, so we actually did look at mortality. I must say that overall, across the whole experiment, mortality rates were pretty low, which didn't give us a lot to analyze. We were very successful in growing these baby trees. However, the trees that exhibited the highest mortality were the ones that were smallest by the end of year 1, which is probably not super surprising to a forester. The smaller you were, the more likely you were to die, quite dramatically so. And in that first year, it was the inoculum treatment that gave this massive growth boost. So putting together the pieces of that story, if you got inoculum, you were much bigger, 50% bigger in the first year. And if you were too small in the first year, you were, as a tree, more likely to die. So that suggests that in sites there's a really high potential for mortality due to climate stress or failure to establish, that the inoculum treatment in particular might be helpful.
Yeah, but more testing actually needed really to then match the soil to the tree type and things like that, which obviously in the case of the Sitka was done, but intriguing area. We sort of answered this already. So there's a question from the University of Glasgow. Great job. Have you looked at the mortality rate? So we kind of touched on that. We didn't really get enough mortality, which is a good thing and a bad thing at the same time, isn't it? And then which nutrient dominates the carbon accumulation in this forest? Bonnie, back to you, maybe?
Yeah, I mean, it's impossible to say definitively without a fertilization trial, which we didn't run, but in most forests of this latitude, it's probably nitrogen that ultimately constrains growth. And nitrogen-poor soils is why these trees have evolved to depend on their mycorrhizae. The soils at this particular site were fairly nitrogen-rich, which reflects their long history as under pasture. And the fact that the trees benefited from the rock dust, which contained calcium and magnesium, but not nitrogen or phosphorus, also suggests that mineral-derived nutrients in forests like this could be important as well.
So, and actually a follow-up question from also from Glasgow. So one small suggestion, maybe in the future, thinking about which species, single species or mixed species plantations should also be considered. So you want more splits, more tests, of course. So all sorts of things that we can work with others, maybe to test on other sites. Obviously, we've got this on the ground already, and it's pretty big and complicated. And if you haven't seen it and you'd like to come and see it, then please do. Give us a shout out, or go to our website and have a look and come to the site, because 72 different test cells, each of those test cells are 400 trees, 100 trees all individually barcoded. It's on a massive scale, which typically sort of people just go, when they first arrive, they didn't realize it was that big. So do come and have a look if you're in the area. We'd love to see you. So what is the sweet spot for basalt dose rate per hectare? Is more better? What about particle size? So I'm actually going to ask Colin to jump in on this one, because we have another parallel study that was set up originally by University of Bangor, master student, which actually looks at different doses was planted at the same time. And I think, Colin, I don't know whether you can remember the results, but you did the analysis. So if you can't remember the results, then I'll go off the top of my head. But do you want to talk to what we found from there?
Honestly, I don't totally remember. I think there was a saturating response.
Right.
Yeah. So more is better up until a point, and then we see diminishing returns. You might be able to achieve the same response with a smaller dose if the particle size is finer, because in theory you should be weathering that rock faster because there's more reactive surface area. But I would guess David is better suited to answer that one than I am.
There's a couple of things here. One is, as we discovered doing these trials, there's a practical limitation to how many times you can reapply the rock. And another way to think about it, if you were doing a reforestation project, might be to plow in a lot or introduce a lot of material at a very high application rate at the start of a project, and then not repeat in subsequent years. I think one of the things that we've done on this project is to repeat once, twice the application rate, and then each time you do it, it's harder to do it because the trees are bigger, the understory vegetation grows, so there are practical limitations on how often you can apply it. So I think there's different ways to think about how you'd manage this going forward. One would be very high application rates at the start, and then don't apply anymore, and then measure how carbon capture changes over time. And the other would be is what we've done here is 30 or 40 tons per hectare, and then try and repeat it 2 or 3 times, and then let the system respond to that rock dust application. I think it's so new that these different techniques haven't really been properly evaluated yet.
Yeah, the challenges of the second deployment that we did, which was after 3 years, were significant, to say the least. We had to figure out how to do it. You've got baby trees, so in effect, you've got a compact tractor is the only thing that can get through it, which we then used a top spreader, typically used for putting sand on a golf course, that kind of thing, and it was very time intensive and difficult to do, compared with a standard lime spreader that you spread the lime at 40 tons a hectare before you do the deployment in the before you even do the ground prep. So much easier to do. The dose response study that Colin and I were mentioning earlier on, we've not published any results from that, but he's quite right. There is a tailing off effect in terms of the growth signals that we saw on different doses. And I think we went from half to 8 times at 40 tons a hectare, so up to 320 tons a hectare kind of level. And you see that tailing off at around about the 40 tons a hectare. So that's a great rule of thumb to think about as a starting point. There's also another piece of work, which again is unpublished, which suggests that higher levels of basalt may then change the water retention ability in the soil. So there is obviously a balance here that needs to be struck. So hopefully we'll get something out in that area relatively soon, and that will help fill in one of the gaps. Right. I'm not seeing any more questions. Heather, you're on the back end. have I missed any? Because I'm juggling lots of screens here. Oh, yep, one more's just come in. Basalt dust can be a waste product. How much waste is available, and is crushing just for trees viable? So we think of it as a byproduct rather than a waste product, as in it has its own value, it has a market in its own right, rather than being, you know, it's a waste product. Sounds sort of something toxic, which is not really the case at all. And there is a lot of it available. We were at the basalt mine up in Bilth, which was our source here from Heidelberg Materials, at the beginning of last week, and there is a lot of this available. So I think the key challenge probably is your proximity to a suitable basalt mine, you know, and then maybe if you've got a combination of acidic soil and you're close to a basalt mine, then this looks like a very attractive opportunity to then, you know, with a little bit of extra effort, to make quite a big impact. Is crushing just for trees viable? I don't think with basalt crushing just for trees is even necessary, actually, because there is so much of this available. David, you looked at this, I know. Any thoughts on is crushing for trees viable?
I think at the moment there's for the UK quarrying in the basalt industries, mostly in Scotland and Northumbria, though as you say, there's some in mid Wales, and they do actually have stockpiles of it. Some of it does go into asphalt, but some of it is a byproduct, like you say, or we could actually call it a climate asset. And I think those quarries are, you know, starting to switch on to the realization that what they've got is a valuable product for improving forestry, crop production, for carbon capture. So I don't think that we're at the point where you need to mine it, especially for reforestation projects. If the government decided that they want to support reforestation as a strategy to help get them to net zero by 2050 and upscale it dramatically, then, you know, you might have to increase production from existing mines. But I think at the moment, there's not that prospect is not really on the horizon.
Yeah. Okay. Any other questions? My last call for questions. I'm not seeing any at this point. In that case, I'm going to thank all of our illustrious panel. Thank you, Bonnie, as well, for your presentation. Really appreciate you being here today. One quick shout out as well to Sonia Winder, who I didn't see was on the list, but actually was heavily involved in the design of the study as well when she was at Till Hill. So really appreciate everybody that's been involved. I mean, this is literally hundreds of people have been involved in this study, and there is this cannot be done by, you know, one or two individuals. This really requires people power. So this is a great testament to what you can do when you put, you know, professional scientific teams together with the right resources and community scientists out in the field who show up, volunteer their time in all weathers, to then go and collect the data, which is essential for these kind of projects. So from me, thank you very much. Really appreciate your thoughts and attention, and really appreciate your support in this study so far.